everybody, to the second in the series of the 2022 Horse Management Seminar presented by Rutgers Cooperative Extension and the Equine Science Center. I really appreciate everybody joining us. We have hopefully going to have a huge crowd tonight. Last Tuesday during our veterinarian, our horse health day, we had over 200 people on the webinar. So it is great that you guys all wanted to join us. I'm going to give you just a little bit of background about what the Equine Science Center is and does, and then run you through about how the webinar is going to work. So first off, I would like to introduce myself. I'm Dr. Carrie Williams. I'm the Equine Extension Specialist here at Rutgers University and work both with Rutgers Cooperative Extension and with the Equine Science Center as their Associate Director for Extension. So instead of me talking all about what the Equine Science Center is and does, we have a very nice video that I would like to go ahead and share for you. So go ahead and take a listen, and I will be back to talk to you in just a little bit.
Good morning. Welcome to the George H. Cook campus, home of the New Jersey Agricultural Experiment Station, the School of Environmental Biological Sciences, and of course, home to the Rutgers Equine Science Center. The Equine Science Center is a soft-walled center where we bring together the best and the brightest to solve challenges impacting horses, horse well-being, and the horse industry. To provide you more details about the research being conducted at the Equine Science Center, and specifically the Equine Exercise Physiology Lab, I'd like to introduce Dr. Kenneth McKeever.
The lab was founded in 1995, and since then it's been one of the most productive research labs in the country, if not the world. We've conducted studies looking at how to care for the aged horse. That's a horse that's over 20. We've looked at various drugs that affect performance. We've even done studies for the Army looking at food extracts that can reduce inflammation. So the Equine Science Center and the Equine Exercise Physiology Lab are a valuable asset to how we care for horses in the state of New Jersey.
So we've all visited this morning the Equine Exercise Physiology Lab, part of the Equine Science Center at Rutgers. We have another facility where cutting-edge research is being carried out, located on Riders Lane, and it is the site of the first of its kind environmental best management practices demonstration horse farm, located here on the George H. Cook campus. The team of scientists who are helping horse farmers become better environmental stewards and caretakers of the land is a team of multidisciplinary faculty, students, and staff led by Dr. Carrie Williams, Dr. Mike Westendorf, PhD student Jennifer Weinart, and other faculty, students, and staff from different departments here at Rutgers and beyond.
Hello, I'm Dr. Carrie Williams, the Equine Extension Specialist here at Rutgers University and the Associate Director of Extension for the Equine Science Center. We are here at the Environmental Best Management Practice Demonstration Horse Farm. This serves as a wonderful venue for the public, farm owners, other industry professionals to come look and see the 8 various best management practices, educational venues that they can use on their farm or in their areas. It is open to the public. You can come anytime. This venue also serves as a great location for research. My current graduate student, Jennifer Weinart, is looking at both cool season and some novel warm season grasses to see the production as well as the horse's metabolism. Some glucose and insulin responses as well as the microbiome will be looked at over the next 2 years of this project. Thank you for visiting all of us at the Equine Science Center. Our website can be found at esc.rutgers.edu.
So while the program that you're listening to tonight is free, we do have a lot of free programs as well. They aren't always coming without a cost to us. And really caring and doing all the research on these horses does come with a cost. So what we would ask you to think about is, even if you have $2 in your back pocket that would like to donate and give back to the Equine Science Center, we would appreciate that. You can go to the esc.rutgers.edu website, /donor, is where you can consider giving your monetary gift. It helps us, like I said, run programs like this, as well as research and many, many other things. So if you're not familiar with the Equine Science Center, I highly recommend you go to the website and check out more about what we do. So what I would like to do next is, now that you all know a little bit about us, I would like to actually learn a little bit about you. So I'm going to share my screen one more time and ask you all to either use your phones or open up another window in your computer, but go to a website. It's really easy, Menti, MENTI.com. So menti.com. I'm going to put it in the chat for everyone. And then I'm going to share another link for you, MENTI.com. So go there, and then I'm going to give you a number that you're going to type in as well, and you're going to answer 2 questions for me about yourselves. So these 2 questions are going to be, here I'm going to start that. So once you get into the Menti, you can enter in the number 66078491. Enter those numbers, and I will start to see people joining us in Menti. And the first question you're going to answer for me once you get in there is going to be, what state or country are you listening from? And please don't abbreviate. And there's an important reason why you should not abbreviate. Spell out the whole state or country. And if you're in the America, of course, you don't need to put USA, just give the state. And then, or if you're in another country, and I do this because we do have some attendees coming in from other countries, and it would be really nice to know from where. So I love these word clouds because as the more people that answer the same thing, the bigger the word is going to be, which is exactly why I said don't abbreviate, because you can see the NJ is actually really tiny, where the big word New Jersey is actually much bigger. So that means more people are actually either from New Jersey or Pennsylvania. So again, as people are starting to log on, please go ahead and go to Menti, MENTI.com, and type in the number 66078491. And you can see that at the top of your screen from what I'm sharing, and you can watch the word change as we start to get more and more people online joining us. And right now, I'm actually looking for some of our international people. I see Canada. Canada's getting bigger. We got a few from Canada. Oh, and even in Ontario, Canada. So that one would be included there. I'm going to give this a little bit until I see things start to slow down. Right now, we have 78 people on the Menti, but I know we have, we're getting close to 150 people actually on the webinar. So, oh, Germany, there's one. Welcome, Germany. Thank you for joining us. But we've got Michigan, New York, Illinois, Georgia, Virginia. It's hard to read the words when they're all moving around so quick. Kentucky, New Hampshire, more Canada. Canada's getting bigger. West Virginia, South Carolina. Wow, there's a lot of different states. Once we're all done with the webinar series, I also like to go see if we've covered all 50 states. I think last year, these webinars, we made 39 states. So we got close. But I don't see as many international people, which is okay. Maybe they're still getting logged on, but welcome. So this will stay open for a little bit, and you guys can continue to log on. But the next, we do have one more slide, because I also like to know our demographic in terms of what you all do. So the next one, the question is, what is your primary interaction with horses? And actually, you can choose more than one. I say primary, but you know, if you're a horse owner and also give lessons, you can pick both of those. Horse enthusiast, maybe you don't own a horse anymore, or you don't yet, you can pick that one. But I like to also watch this as the bars grow and change. If you want to, if you said other, I know we have a couple other things going on. If you don't mind sticking in the chat for us, maybe tell us what other. Are you a farrier, a massage therapist, involved with therapeutic riding? There's a lot of different things. So I do see we have a farrier. Welcome. Thank you for joining us. Extension agents. Awesome. Thank you. I love my extension agents. But it looks like we have most of you are horse owners. We got a lot of enthusiasts as well, doing training and lessons. And a big other category today. We actually didn't have a very big other category last week. You lease a horse. Okay, good. I would say that almost is horse owner. You still got to pay for a lot, right? We got students, a judge. Excellent. Welcome. More students, an equine science student from another university. Welcome. A therapeutic riding instructor. Great. A path instructor. So a lot of therapeutic riding instructors. Oh, great. So welcome from Germany, a state advisor for the horse husbandry in Germany. Excellent. So this is exciting. I love to know where everybody's coming from and what you're involved with, logging on and things like that. So let me, while we're letting this populate, I'm going to go through just a couple different things or housekeeping tips, I guess, before I introduce our first speaker. And I know I see some of you found this already. You found the chat. Well, that's great if you need to reach out to any of the panelists for any reason. However, I would say let's not use those for any questions you have for the speakers. What we're going to do is use the Q&A box. And the Q&A, once I stop sharing, if you go ahead and kind of shake your mouse over the screen, you'll be able to see a little Q&A. It'll have two little bubbles on it pop up. And you can type any of your questions in that Q&A box. And that way, as the speaker is talking, I can either go ahead and moderate them, or I can flag them as to save for the live Q&A at the end. Or if I might know the answer, or I might want to answer the question myself, I'll be doing that while the speaker is talking, but everybody will be able to see the answers. So you can go ahead and do that as well. So I'm going to stop sharing. Thank you guys for playing with the Menti for me. And then I'm going to go ahead and ask Dr. Brooks to come on the screen and start to share her screen while I give you a little bio of who our first speaker is going to be. So welcome and thank you, Dr. Brooks. Dr. Brooks is a lifelong horsewoman. She was diverted to vet school by a budding passion for equine research. Following a bachelor's in agricultural biotechnology, Dr. Brooks remained at the University in Kentucky, so that's where she was, for her PhD in veterinary science. She specialized in equine genetics. Following her PhD, she was awarded a postdoctoral fellowship to study the expression of inflammatory genes in horses affected with laminitis. So definitely a big hot button topic there. From there, she was an assistant professor at Cornell for 6 years, but now she's an associate professor at the University of Florida. Her research group has discovered genetic mutations and markers for coat color, height, sarcoid tumors, and 2 different neurological conditions. Her ongoing work targets variation in gait, susceptibility to infectious disease, metabolic syndrome, and skeletal defects using a wide genome-wide association and genome resequencing and transcriptomics. Oh my goodness, you're putting me to the test today with all of these words. So you can tell my field is not genetics at all, but I heard Dr. Brooks give a very similar and amazing talk this summer. I loved it so much I had to invite her back to share her expertise with all of you. So she's going to be talking to you today about genomics in the horse industry, discovering new questions at every turn. So
great. Thank you for that introduction, Carrie. I'm just going to say hello to everyone because when I start to present, you won't get to see me anymore. But yeah, I'm going to tell you a little bit about some of the new innovations that we have and some old innovations, truthfully, in genetics and genomics, which is the study of the whole genome, that are very relevant to the horse industry. But I found that a lot of people are just not aware of all the things that we can do with horse DNA. So let me try to share my screen and we will get started. All right, so the fun thing about working in horses is that we do have a brand new horse genome sequence. Our horse genome sequence was completed in 2008, but published here in 2009. And that sequence came from a thoroughbred mare named Twilight. This is Twilight here. She lives at the research farm that's managed by Dr. Doug Anzac and is a part of his cornerstone research program on immunology. A genome sequence gives you all the base pairs in the nuclear DNA that contains the instructions for making proteins that do all sorts of the processes of life. So genes for things like making pigmentation, for making long legs, for making a hardy constitution or a strong immune system, they're all coded for within that genome, along with some instructions on the how and when and where they might be used. So the neat thing about a genome is it's not just for horse breeders or for use in selecting matings. There's a lot of other fun applications. So once you have, scientifically speaking, that whole genome sequence, you can do things like make custom antibodies for assaying all sorts of interesting proteins. So the genome itself is catalyzing lots of research outside of just genetics. It also does things like give you a catalog of all of the proteins encoded by that genome so that you can go through and use protein sequence to identify what might be happening during a certain disease or a difference between horses. It also, of course, gives us a catalog of expressed genes. So this is nice because we can use this to track what a cell is doing at any given moment during a process. So for example, we just published a paper looking at the entire transcriptome, or all the genes that are being transcribed in laminitis itself, and discovered some interesting inflammatory pathways that without a whole genome sequence, we never would have been able to investigate. And the fun part is that then genomics can go on to inform our management. We, in production agriculture, use terms like precision management. And in human medicine, you talk about personalized medicine. But all these fancy words mean is that we look at the genome, identify conditions and situations that might predispose that horse to illness, and then make targeted decisions to try to avoid or intervene. One example being nutrigenomics, which we might talk a little bit about later. And of course, it is an old standby for selective breeding in many different species, although I would say it's currently underutilized in the horse industry, partially just because most folks don't know about the variety of tools that they have available to them, but also I would say because our horse industry is pretty wed to the mystery and art of matching sires and dams to create that potentially exciting next generation. So when I talk about some of these genomic tools, what I'd like you to keep in mind is that it's not just for the horse breeder, that as we begin to produce new tools with more forward thinking and better predictive abilities, we can apply them not just to diagnose disease or diagnose potential for excellent performance, but also to incorporate in your day-to-day management of your horses. Horses are a long-lived species. I think everyone out there knows that they are a little bit expensive at times. So the idea that we can improve their quality of life and improve the economic efficiency of our operations through genomics, I think is really exciting. And hopefully by the end of my talk, I may have convinced you that using genomics, it can be a lot cheaper than not using it at all and getting some surprises. Okay, so here's a quiz. Now, I can't see what you all are each doing because you don't have your cameras on, but on your honor, no cheating. I want you each to sit and think just for a moment, how many genetic tests can we utilize commercially today for the horse? Or how many do you think that might be? Just scribble it down on a little note paper, maybe write it on your hand. We think it might be 5 to 10, 10 to 100, 100 to 1,000, or more than 1,000? Certainly in human medicine, we're well over 1,000, right? Well, my best estimate, which is always changing as new publications come out every day, is that there are at least 100 to 1,000 traits that we could test for in the horse, depending on how you count, because some of these individual regions of the genome actually contribute to multiple traits. The challenge is that there are some speed bumps in translating some of these tests into industry. There's some challenges in terms of economics, lots of work left to be done in extension to help horse owners understand what they're looking at. So we're getting to where these can get into the hands of the people who can use them, but there are challenges left to face. I hope you got that right. My students will tell you I tend to like answer C. I'll try to mix it up a bit as we go. So I mentioned earlier that as horse people, we are a little bit in love with the art that is horse breeding. And generally, when I poll horse owners, most of them can actually quote to you more generations and ancestors on their horse's pedigree than they can on their own, right? So pedigree questions are often central to the horse industry and can significantly impact the economic value of a horse. So one of the first little research vignettes I'm going to show you is actually about the pedigree and what can happen there. Before we get there, though, I'm going to take us back even farther, because one of the neat things, so pedigrees are human history, and we are very much in love with the pedigree, right? But there is so much more to learn about the horse before we started keeping the pedigree. So this first sort of third of the talk, we're going to talk about what happened when horses met humans, using the DNA to tell the story. And then we'll come back to modern times and examine those paper pedigrees that we are so much in love with. One of the neat things about the horse is that as a species, they were extremely successful. And equine fossils are some of the most prevalent fossilized mammalian remains on the face of the earth. They are just incredible creatures and have been for 55 million years. So up until this spring, the world record for the oldest sequenced DNA genome was held for the mammals by a horse fossil. And this was a horse who lived almost 750,000 years ago from this tiny fragment of bone that came from the permafrost up near the Bering Strait in Alaska. That is a lot of history and a lot of biology that we can learn from. Based on archaeology, our studies up until very recently indicated that domestication of the horse first occurred in Kazakhstan a little over 5,000 years ago. But we know very little about the horse that they used at that time. The way they documented the chances that these were domesticates is that they could find evidence of milk residue in pottery, and chances are if you're milking it, it's domesticated because wild animals are not fond of being milked. And they could see what evidence of what they thought was likely bit wear on the front teeth of the skulls and these deposits. They sort of drew an analogy to what some of our dental professionals do to the teeth of riding horses today when they create a bit seat by smoothing off those premolars so that the bit is less likely to catch or jar on any points at the front. I think they can accept that these horses were probably domesticated. What else can we learn about them, and why should we care? I'll get to that point too. So some of our colleagues who focus on ancient DNA were able to obtain bone samples from these horses located here in Kazakhstan, where they were buried with members of the Botai culture of people about that 5,000 year ago mark. They used the sequence of those genomes compared to other modern horse genomes and some horse family members like the donkey to calculate how much time had likely passed since those ancient horses last shared an ancestor with the other members of the groups that they had to compare to. So they compared to donkeys, and on this little family tree, you can see that the branch length is very long, so donkeys were a distant relative. Other ancient horse samples were also fairly distant. And when they looked at modern horses here on the right with a chromosome number of 64, they could see that actually these early domesticated animals were not a good match. Instead, these first domesticated horses were a close match to the modern Przewalski's horse, the Mongolian wild horse, which fortunately their numbers have recovered, but they were quite endangered there for a while. And they are completely a different species with a chromosome number of 66. So while we were riding them, they were not the ancestors of all of our modern horses, which left us with a big research question. We know that the two branches of this family tree separated about 45,000 years ago. And we know that somebody was riding the ancestors of the modern Przewalski's horse about 5,000 years ago. But what happened in the last 40,000 years to create a relationship and a new domesticated species that has been so central to shaping human culture? Well, it was exciting news at the time, completely rewrote the textbooks. I had to change all of my lectures to say that horse domestication did not happen 5,000 years ago, and we as scientists have absolutely no clue what's going on. And it did win a lovely cover on the Journal of Science magazine, which is a bit like being Time magazine's Man of the Year, something like that when it comes to science. So this is definitely the Rolling Stone magazine for nerds. So fortunately, our colleagues did not give up there, and they continued to use the genome to better understand where this modern domesticated horse might have come from. The answer was really quite interesting. By mapping the relatedness of an additional set of 273 ancient horse genomes, they were able to identify a region just north of the Caspian Sea, where the vast majority of our modern domesticated horses can apparently trace their ancestry. They also, based on some molecular clock type techniques, can time the emergence of the modern domesticated horse to about 3,000 years ago. That doesn't necessarily mean that that's when domestication occurred, but that is when we all selected the horses that we have primarily kept with us today. This raises some additional questions, though, because about 3,000 years ago, we did not have any major turnover in human populations. There were no huge migrations, no large wars. This did not correspond with our big historical events like the conquest led by Genghis Khan, things like that. So why, at that time in history, did humans abandon all the other horses they likely were riding and using in this ancient domesticated and in that Botai Przewalski's horse line, and all of a sudden switch to an entirely new type of horse? Well, luckily, additional research on those genomes can tell us a little bit about the why of this question as well. And my slides are in a hurry to move on. In fact, the genome shows us that there were only two regions of the genome that were key in this change. And here is why I'm going to convince you that you should care about ancient genomes a little bit, because up to this point, you're like, it's old history, Brooks, move on, right? How is this relevant to the horse in my backyard? Well, let me tell you. These two regions of the genome are the primary changes that drove a drastic shift in the horse population around 3,000 years ago. And when we study the genes that are encoded within these regions, by looking back at our whole genome sequence, we found some very interesting functions. For example, at this one first locus on chromosome 3, that gene is particularly important for controlling issues of anxiety and aggression in human pathologies. And when we look at the other locus, the green one that I cannot manage to keep on the screen for some reason, that one is actually mapped as contributing to spinal stenosis and back pain in humans. So you can imagine that changes in these regions probably contributed to things like docility and hopefully less back pain. And I can just about guarantee that everyone out there who has interacted with horses has known a horse that might have had a challenge with temperament or aggression. And statistically, you've all interacted with a horse who is likely affected by back pain, potentially from a condition called kissing spines. The interesting thing too is that these two loci are not yet fixed in the genome. So our collaborators and I are currently working to better understand those genes and to see if they may still be shifting within the population of modern domesticated horses. It also has fueled a bit of an inside joke in my lab when I say, okay, why did they shift horses 3,000 years ago? Well, the interesting historical coincidence is that those bow tie people who were attempting to ride Przewalski's horse, not only did their horses nearly go extinct, but the bow tie culture went extinct as well. And our inside joke is, well, it's because they were trying to ride Przewalski's horses and they all got bucked off because they all were mean and they had back pain. Of course, we don't know if that's true or not, but it certainly helps us to sort of picture the relevance because my lab is populated by a bunch of horse folk and most of us have been bucked off. So we'll keep working in that area to see if we can learn more, and hopefully we'll develop tools that can be used for diagnosis and selection, maybe to improve things like temperament and back health in our performance horses. So let's shift gears a bit. I'm going to bring you back. We started this story with the modern pedigree. Let's come back to the modern pedigree and how we feel about that. The interesting thing about horses is that we have kept extensive records. It's a cultural phenomenon to us as humans. And for the horse specifically, a lot of these records are exceptionally deep. We often like to quote that for some breeds of horse, like the Arabian, that we can potentially go back as far as records that are 900 years old. I don't know how documentable that is, but we definitely do better than we do for other livestock species and companion animals like dogs. And for a geneticist, this is a potentially exciting set of records. The neat thing, though, is that while pedigrees are history as recorded by man, the DNA is the history and ancestry of that horse as recorded by the horse itself in every individual cell. So if you ask me which record I want to look into, I want to go to the horse's mouth, so to speak, and hear what biology has to say, a little less about what humans have had to say about the ancestry of that horse. So I'm going to take you back to your high school biology class, maybe even earlier than that. Let's take a quick quiz again. Think of two full siblings, and I've got two horse siblings as an example here, but you also could think of your own siblings. How much of your genome do you think you share with your full sibling, or would a horse share with its full sibling? This is a key question, because if you're buying that horse based on a pedigree, those two full siblings have completely identical pedigrees. You cannot get additional information there to distinguish the two. You have to move on to other things like visual examinations, performance records, and so on and so forth. All right, so do you think it might be 10%? How about 25%? Sorry, that's for half siblings. Full siblings are actually 50%. My annotation lied. I told you I like answer C, right? Full siblings share about 50%, half siblings about 25% of their genome. That means they can differ by almost half of their genome, depending on how deep that pedigree is. So 50% is the right answer here. I thought I would change things up as I was drawing my slides up. So just as a proof of concept, I'll show you a little bit, a small piece of data here based on some Arabian horses. This is an example we use for some of our pedigree analysis there. If we had a horse, here's just a caricature of a horse, that we will put in a set of markers typed from the DNA. So in this case, this study used about 50,000 different sites within the genome where we know that there is a difference among horses. So there's a little place where there's data where we can detect diversity. And we will put these markers into a computer for any number of horses and ask the computer to infer where are the strong similarities and differences across this population of animals. Now, this is really cool because that means it is assumption free, so to speak. So I don't tell the computer what I anticipate to find. I let the computer examine the data and then tell us what it finds. And then we have to give it a little bit of historical interpretation to try to better understand it, maybe. So for this example horse, his ancestry can be attributed about 50% to the blue group, about 25% to the yellow group, and about 25% to the red group. So a real life example might be a warm blood horse who would have perhaps 50% of its ancestry attributable to thoroughbred ancestors, maybe 25% to Arabian, and maybe another 25% to heavy horse. Do you get the idea? Here's another sort of illustration to kind of help you. So this is some perceptual art that I kind of like to think of, in that imagine our 50,000 genetic markers are just thrown up in the air and allowed to just assort themselves based on the relationships between the horses. And when you first do that, it looks a lot like this art where it's kind of a jumble. You really can't tell what you're looking at. But the computer can examine those relationships and find within those seemingly unrelated data points the common themes that allow patterns to emerge that hopefully we as humans are able to interpret. So this art, if you just walk around this piece, it changes the point of view and allows the pattern to emerge strictly from the data. So let me show you some data from a large study we did on Arabian horses, in part because Arabian horses have deep pedigrees, but also because as an industry, they were highly motivated to learn more about their ancestry. So this study utilized just shy of 400 Arabians from 12 countries around the world, and we compared those to reference groups, including 100 horses of Persian ancestry and 400 horses from known breeds. We did generate 14 whole genome sequences as part of that project, which these days is a drop in the bucket. And in this case, we started with 300,000 genetic markers, although we slimmed that down quite a bit as we filtered for quality. And if you're interested in reading more about this project, the nice thing about this particular journal is that it's open access. So anybody can Google that title, click on the link, and see all of the images and the scientific report of what we did. It is a little dense, but I promise you, dive right in, and if you're interested by ancestry and Arabian horses, you will find some fun tidbits there. So think of this plot a little bit like that perceptual art, in that each dot is an individual horse. It's a bit like a three-dimensional family tree, but we're just looking at it from one side. It's colored by breed. So the deep purple are our Arabians, which cluster together as they should. At the bottom here, we have the thoroughbreds. They've had a long history of a closed stud book, so they tend to draw away from the horse family tree more. And then on the top here, we have some of our heavy horses, like our Belgians and some draft breeds, as well as the Icelandic, that have their own characteristics. So it was nice to see that the Arabians had a distinct signal, but they also had a lot of diversity here. So the lesson to learn is that while Arabians tend to be grouped as a single breed, within the breed, you find animals suitable for diverse purposes and with a lot of genetic diversity left to measure. The other interesting thing we did was examine that family tree comparing only that Arabian group, so we could look more closely at the diversity. And here we could see some interesting trends. So we included the thoroughbred as an outgroup group. They're up here at the top. And the straight Egyptians, actually, because they're a fairly unique lineage, stuck out a little bit to the right. But then we had some interesting horses that fell a bit close to the thoroughbred cluster. And so we thought we would look into it a little bit further. All the horses here not marked as thoroughbred were fully registered as Arabian horses by the appropriate registry in their country of origin. Now, this amazing rainbow works exactly the same way as the column I showed you earlier on with our example horse that had blue, red, and yellow ancestry. Each line in the plot is a horse. Blue is still thoroughbred. Here, for example, Icelandic is the light yellow, and as it happens, red in this situation is a Turcoman ancestry. The neat thing I want you to examine here is that Arabians on the left have a lot of genetic diversity there, a lot of deep history not documented in their pedigrees. But we found some surprising things as well. For example, we found a lot of color sharing between thoroughbreds and modern flat course racing Arabians, which was not documented in their pedigrees and created some challenges. We could also document some old sharing between thoroughbreds and Turcoman and some of the rarer lineages within the racing Arabian, which might represent some of the things we do know of historically, thinking of those three cornerstone thoroughbred stallions, including our Darley Arabian and our Byerly Turk. They might be right here in these signatures. So given the somewhat surprising finding we investigated it further using another pattern-seeking piece of software. And in this plot, each line is a horse. So we've got a set of horses here. And the yellow is the Arabian signature, whereas the blue is the thoroughbred signature. So we could see among racing Arabians, some horses that really were primarily Arabian, and some blue would not be surprising. You can see there's some green in here as well, right? So shared ancestry with Icelandics is not well documented, but they're all horses. So you go back a few thousand years and I'm sure they did share an ancestor, right? What was surprising is that this bottom group included horses whose genomic documented ancestry was up to 2/3 thoroughbred, which didn't agree with the pedigree at all, and may create some challenges for that registry. So they're still looking into this a little bit, but none of this was documented by the pedigree and created some hard feelings among horse owners who didn't feel that they had gotten a horse or a stallion that was accurately advertised. Of course, the motivation to do this might be obvious in that the thoroughbred was more strongly selective for performance on the flat race course. So potentially they have a performance advantage on the flat course racing, and folks wanted to win. So they found a way to do it. We did also look at the Y chromosome to think about that lovely story of the Darley Arabian, the Godolphin, and the Byerly Turk. And we could see that not only are their signatures apparent in the thoroughbred group, but also that the 2/3 of that Arabian pedigree we found in the racing Arabian was not ancient, was not 200 years old, but no doubt happened less than 200 years ago based on the sequence of those Y chromosome markers. And we did not see any evidence for this type of admixture in non-racing male horses. We only identified this signature in racing Arabians, which strongly suggests that at some point the new ancestry was introduced by stallions primarily, crossed onto Arabian mares. So industry perspectives, this created a little bit of a stir. As scientists, we like to find unexpected things, but as humans, sometimes we don't like surprises. So we did get a fair bit of media coverage for this particular story. Everything from, oh, of course we knew that because they look like thoroughbreds, to some posts and stories that actually impugned the reputation of the scientists who participated, saying that we must have faked the data. So it's quite a mix, and we don't know yet what kind of regulatory complications may occur due to this. It's not necessarily our job as scientists to make decisions there. We study the data and we report it, and then it's the responsibility of the horse industry to figure out how they're going to deal with that. All right, so let's think about some of our more practical everyday tools. So horse owners, you can buy ancestry testing for your horse You can learn, you can get reports about those colors and those bars. It's lots of fun to do it in warm bloods and sport horses because we're always interested in the proportion thoroughbred blood, especially in our event horses. But the way that most horse owners are introduced to genetics and genomics is usually through individual tests. So I want to examine those a little bit and specifically think about, whenever that domestication event actually occurred, what are some of the costs? So we saw where 2 places the genome shifted drastically just at that ancient time point. But at the creation of breeds, whenever we start to select for some common goal, we often get some fairly drastic changes. One thing I want to point out is that we all have alleles for things that are considered recessive diseases. So when I first started writing this type of talk, it used to be estimated that each human carried 3 disease variants. Now it's estimated that each human carries between 40 and 110 disease variants. Most of us, fortunately, are healthy because we frequently don't practice inbreeding and line breeding among most human cultures, not all, but most. And that diversity and broad gene pool has helped protect us from some of these recessive disease alleles. It's not always true of livestock and horses. Sometimes for the better, because we would not have characteristics that are consistent across breeds if we didn't practice selective breeding and some inbreeding. One story I want to use as an example case is that of hyperkalemic periodic paralysis, or HYPP. I focus on this condition, well, because it's not rare, but also because it was the first genetic condition for which we had a decisive genetic test. It was developed way back in ancient history in 1992. It is incompletely dominant. That just means that if you have one of your 2 copies has this genetic change, that you will see some health-related signs. 2 copies in this case is even more severe. So affected horses, they have a change in a sodium channel protein resulting from this genetic polymorphism. They have muscle fasciculations or twitches and can lose control of that muscle to the point where they may not be able to stand. And of course, your heart is a muscle and your diaphragm needs muscles to power your breathing. So it can be potentially fatal. And when the disease was first discovered, the fatality rate was estimated at somewhere around 2/3 of horses by the time they reached 6 or 7 years of age. The other interesting thing to note here is that it occurred in a very prominent quarter horse line. As geneticists, we don't like to name names, but it's now broadly known that the horse named Impressive was common among all of the carriers of this disease at the time it was studied in 1992. At that time, he was around 24 years of age and was estimated to have perhaps over 50,000 progeny. So he was sort of it was his popularity that perpetuated this allele as much as anything else. Now, it's now known that he was not the only horse to have that allele at that time. He was just the most popular. And like I just said, 40 disease variants per animal. This just happened to be his one of 40. But any other animal that gained popularity like this probably would have also had a disease associated with it. Excuse me. Pardon me, got a frog in my throat. Now, the interesting thing at the time is that with the creation of a genetic test, the authors were so bold as to propose that the genetic test provided an opportunity for control or eradication of the condition, which is kind of our, motivations for doing our work, is we hope it will help to control or eradicate disease, sometimes treat it. So let's move on to another quiz. At least 20 years have passed since we developed the HYPP test. Take a little note. Do you think that the frequency of HYPP has increased, has stabilized, or has decreased, like the scientists predicted? Or do you have absolutely no clue? That's a passable answer as well. Do you have your answer? All right, well, this time I did not go with C. And in fact, the frequency of this disease has done the exact opposite of what the scientists predicted. It has gone up. This is despite efforts by the industry to help to control it. So as of 2007, the AQHA voted to no longer register horses who carried 2 copies, but they still permitted horses with 1 copy. And remember, as of 2007, you had 15 years had passed since you had that genetic test for the disease. In this 2009 paper, they measured that increase. So we've not had work done since 2009 in a good broad context. So honestly, it may be continuing to go up. But when it was discovered, about 22% of halter type quarter horses were affected. Now, more than half of halter type quarter horses are affected, meaning they have either 1 or 2 copies of the HYPP allele. So we've really been very efficient. As a scientist, it's my nature to ask why. So why this trend in the allele frequency for something that when it was discovered was considered to be potentially lethal? Well, there's 2 examples or hypotheses, proposed theories as to why this might be true. The first is general market forces. So the value of having a pedigree that had impressive as multiple ancestors within it simply exceeded the loss of value that resulted from the health deficits and potential death of homozygous affected foals. So with no broader market force to reduce breeding of that allele, it just continued to be perpetuated. Horse owners and business owners in particular are often very short-minded by necessity. They need to focus on what will keep their business open over the next year or 2 years or 3 years, and don't always have the luxury or foresight to think about protecting the gene pool of the next 20 or 30 years. That is often perhaps the responsibility of horse registries. The other thing that occurred is that by knowing the change, that sodium channel that was altered, we particularly modeled our management of this condition based on treatment of a human condition that had a change in the very same gene. So we learned, especially through nutritional changes, but also through general management practice, like avoiding stressful trailering and making sure that we had sufficient electrolytes in order to mitigate a lot of the negative health effects of the HYPP allele. So that's kind of a good thing. Horses die from HYPP far less frequently now than they did when we first discovered the genetic test, in large part because the genes told us how to change our management and nutrition to better control it. There's another possibility, though, because there are perhaps 100,000 protein functions in the mammalian genome, but only 20,000 coding genes. So each gene kind of acts like a Swiss Army knife with multiple purposes. And there may be a multi-purpose nature to the HYPP allele. Some hypothesize that a mild version of the fasciculations may actually assist in development in musculature and conformation and shape and appearance of those muscles that for some reason we find appealing. And in one very small study, they considered half siblings, some who carried HYPP and some did not. And they did in fact identify a statistically significant difference in the number of halter points earned. So it may be an example of something we call balancing selection, where one version of the allele is negative, but the other version carries something positive that we're selecting for, which tugs along the disease as part and parcel of that genetic package. We don't have conclusive understanding of what's going on in this situation yet, and I hope that we'll get the opportunity to do some further research to know for sure. Here's another example. For those of you who work with sport horses, you may have heard something about this in press recently. This is a condition called fragile foal syndrome. It affects an enzyme that's important for collagen, and a collagen is the matrix that gives skin its strength. So when this is dysfunctional, you get thin skin that tears easily, resulting in large open skin defects after birth, just from the stresses of moving through the birth canal. It is recessive and considered lethal. So recessive means it takes 2 copies to see this very drastic and catastrophic health defect. And no horse who's homozygous for it has survived to date. They have all either died close to birth or needed euthanasia. Breeding horses is expensive, right? And you invest a full calendar year nearly in getting that foal to term and on the ground. So the economic and emotional costs of this disease are very high. So a few studies launched at first wanted to assess how many affected foals there might be. So just to kind of show you how delicate the skin is, on the right-hand side, you see a little strip of skin from a normal foal on the bottom and the fragile foal on the top. The other thing is that although the disease was first documented because we had some young foals born with some very striking deformities like those that you see here, including the fragile skin, as well as some spinal curvature problems and some angular deformities in the limbs, they also, in a German study, identified that the allele could be found homozygous in tissues from lots of abortion and stillbirth cases. So while a few of these very catastrophic near-term births sort of caught our attention, in fact, it probably had been hindering reproduction in these populations for a number of years by reducing the number of live births and basically going unnoticed. This group in Hanover actually measured the frequency in the Hanoverian breed and found that about 14% of that group were carriers. And they tried to identify some common ancestors that would have explained it a bit like impressive. His common ancestry explained the proliferation of HYPP, but I don't think they actually landed on that answer. The other interesting thing that we've learned through comprehensive testing is that although this disease was originally termed warm blood fragile foal, I'm really not a fan of that name because in fact, we see the same disease allele in diverse breeds. So in the stock horse breeds, it circulates at about 1%, fortunately small. And we even see it in other rare breeds like the Haflinger. So why is the percentage so high in sport horses, which it ranges from 7% to nearly 20%, depending on which population you examine, and is still fairly low in some of these other groups? The other bit of controversy here is that the industry immediately wanted to know, well, okay, so 2 copies of this gene are lethal. What about horses who carry 1 copy? They're fine. We've not noticed problems. They have to be just fine. Well, to answer this question, we don't have sufficient research funding to actually study the horses yet, though we're working on it. What we can do is draw some analogies from human conditions that are similar. In fact, there is a human genetic change in the same gene, the PLOD1 gene that's important for collagen. It belongs to a set of syndromes called Ehlers-Danlos. That's 2 doctors who discovered it, and there are, at least a dozen different Ehlers-Danlos syndromes, and I'm probably underquoting that. People who have this change, they do have some other challenges. They can include problems with their eyes, with their spine, and with their blood vessels. And in severe cases, they can have a catastrophic event like a rupture of your major blood vessel, the aorta, or a stroke. They also have some very interesting conformational features. So people with this allele tend to have increased height. They're taller. They have a somewhat narrower rib cage, a narrower chest, and they're hypermobile. So they can bend their joints with more elasticity than the average human. So that's what this picture is showing you here. Each of these folks, if you just try to take your thumb and fold it back to your arm, you should find that fairly challenging unless you have hypermobile joints. We don't really know well for PLOD1 because it is a fairly rare form of Ehlers-Danlos if we can fully call it recessive. We do see that humans who have this allele have less of that normal PLOD1 enzyme. So the 1 copy they've got is limping along and only making half as much enzyme. What we don't know fully is how many of these structural challenges will be incurred when you only have half as much enzyme. It's kind of like when your car has a flat tire and you throw that donut on there, that little tiny little spare tire. Maybe you can get by at 35 miles an hour, but I wouldn't go try to hit 75 miles an hour on the highway, right? So these people might be just fine as average everyday people, but if they try to go become an Olympic sprinter, they might encounter problems. The industry also reacted to this with remarkable intensity just a couple years ago, beginning in 2018, following an announcement from a very large breeding operation for sport horses, where they openly disclosed that some of their top stallions carried this allele. What's ironic about this is that we as geneticists had known about this allele for more than a decade. We didn't know how broad the distribution was at that time, but we all kind of saw this coming a little bit. There was a strongly expressed opinions, some breed groups stating that all horses who carried the allele should not be registerable. Other breed groups firmly taking the stance that carriers are entirely benign and there's no reason to panic. The truth of it is we didn't have enough scientific information, although we are working to expand our knowledge here. So this is some data very recently published, actually just in January, I believe, by some of our colleagues who were looking at Swedish warm blood performance horses. So remember, in humans, we see improved elasticity of the joints, slightly taller height, and that interesting narrow chest phenomenon. This group compared the genotypes, which in your plots on the right are sorted from 2 normal copies on the left, 1 warm blood fragile foal copy in the middle, and then hypothetically, we've got a difference of between those 2 on the right-hand side. Of course, you don't see the 2 copy fragile foal genotype because those horses have all died. When they examined the inspection records of these horses, they discovered that even comparing within sport disciplines, the carriers for fragile foal had on average higher scores for dressage suitability, higher scores for the quality of the canter, and some significant changes in other qualities like their walk. This was less noticeable in the show jumping groups, in part, I think, because those horses are valued for different reasons, but very significant in the dressage subset of Swedish warm blood. So this suggests that the reason the allele frequency might be so high is in fact because we have another example of balancing selection, that there may be some hidden benefit to possessing 1 allele for a fragile foal. This is some work we're actually continuing on here in my own lab, where we're using some artificial intelligence technologies to examine videos of horses at the trot to see if we can actually measure, are there changes in the joints, or perhaps were these results just reflective of the populations of horses that are more suitable for dressage and maybe bringing along the fragile foal allele as a coincidence. But it was a very thought-provoking kind of piece of work published in January. So with all of these multifaceted concepts to keep in mind, what should the horse owner and breeder, and particularly our horse breed organizations, think about? Should they want to try to manage genetic disease? Well, I hope you have noticed through these examples that definitely takes some consistent strategic and organization level policy to make any impactful change. Clearly, prior to 2007, hoping things will work out on their own was not doing anything to control the frequency of HYPP for the American quarter horse. And while sires do have an oversized impact on the gene pool, if you're a small operation with just a few mares, it's going to be far more important for you to manage that gene pool of your mares as dams of the future generations than just to consider sires alone. So really, to be effective in the industry, we need to test both the male and female paternal lines. And from a practical standpoint, the witch hunt kind of drastic action calls that came out in response to both our pedigree revelations and news about fragile foal are probably not the smart choice. It's something that, from a practical standpoint, likely needs to be phased in with policies that can adapt to meet the needs of the industry and balance both the positive and the negative aspects. So, for example, if all carriers of fragile foal had been blocked from registration a decade ago when that allele was first documented, we would have lost some 20% of the top dressage horses in the world. And that likely would not have been an equal exchange for protecting the loss of the very tragic and inhumane loss of foals with torn skin. And we got to think about a couple generations down the road. So think about controlling carriers as we go forward. Most of all, though, I hope I've pointed out to you that, you can imagine if you're an owner of 2 or 3 of those sport horse mares, sometimes losing a breeding season and having a catastrophically sick foal can push your operation right over the edge in terms of economic viability. So by and large, if we were to take the stick our head in the sand and ignore things approach, these alleles could rapidly increase to the point where many of us are feeling the impact in terms of higher veterinary bills, more catastrophic foal losses, and potentially loss of value of our animals. So at the end of the day, not only does it make ethical sense to utilize genomic tools in managing your horses, but it also definitely makes economic sense. Okay, so let's review very quickly. Where did the horse come from? Well, at this point, darned if I know. It looks like, based on very recent data, it was just north of the Caspian Sea. And that set of horses had 2 features that made them vastly superior to all the other domesticated, per se, horses at the time. And those features probably impacted things like rideability, with the potential impacts from pathological back changes and temperament. At least so far we think so. And next week when a new paper comes out, I'll have to change my slide set all over again. When looking at a modern pedigree, keep in mind that it's an estimate. Now, excluding examples of obvious fraudulent pedigrees, you only have that 50% shared by identical by descent for full siblings. So you need to use something more powerful than a pedigree, and I hope that the genome will start to take that role very soon. Finally, things that are considered a genetic disease are often not simple. Even if they are attributable to a single genetic change, there's an important trade-off to consider, sometimes between health deficits and potential performance benefits, but sometimes also in the economics of what we would like to do and what is practical to do, but still maintain the viability of our horse industry. And finally, I hope I have convinced you that our genomic suite of tools have a lot to offer, but educational activities just like this one, where we get to talk to horse owners, are key. So, because there's only very, very few of us who specialize in equine genomics, and it's pretty tough for us to get the word out to everyone. One last quick note, to try to get that word out, I do have an online short course open to the general public. It has an action-packed set of modules and short educational pieces where you can listen to me from your computer in the comfort of your own home, and learn all the way from the basics of genetics up to some early applications. We actually have now a suite of 2 courses, Horse Genetics 1 and Horse Genetics 2. And as we evolve our online teaching post-COVID, I will say these courses are continuing to grow and to change. And I hope that once things get back to a little bit more normal, we'll get to expand on these and refresh them and keep them helping to spread the word.
Well, thank you, Dr. Brooks. That definitely sounds like a class I should be taking as well. So, but I always learn so much from your talks. And I said last week, and I'll say the same again, I think that the biggest problem with virtual seminars is you don't get to hear the big round of applause when you're done speaking, or hear it, see it, and see people's faces. But But thank you so much. That was great. You do, and if anyone does have any other questions, and you might have missed me say this earlier, there's a Q&A portion at the bottom. So if you wiggle your mouse over the bottom of the screen and you see the Q&A button, go ahead and you can ask our speaker. I'll just ask Dr. Brooks to maybe peek at a few of them. We will save some for the Q&A panel afterward. But if you see any that might be just kind of a quick response in the chat or in the Q&A, please feel free to do so. But thank you again. So with that being said, I'm going to ask Dr. Adams to come on, and I'm going to go ahead and she can start sharing, and I'm going to go ahead and introduce our next speaker. Both very, very good topics that I always like to learn more about. I asked Dr. Adams to come on. She is the Associate Professor of the Gluck Equine Research Center at the University of Kentucky. She's also an adjunct faculty at Lincoln Memorial University College of Veterinary Medicine, and has recently been honored as the Mars Equestrian Fellow, specializing in care of senior horses. She completed her PhD in veterinary science with an emphasis in equine immunology from the University of Kentucky, and then completed a postdoctoral fellowship in areas of immunology and endocrinology. Currently, her research program encompasses the study of equine immunology and endocrinology in areas of aging, obesity, endocrine diseases, laminitis, and stress. All very good things that I would love to be an expert in. Her research program is world renowned and has established and support of unique herd of aged PPID, obese metabolic syndrome, and insulin dysregulated horses for both basic and applied research to further the knowledge in these areas and to prove the health and well-being of aged horses. So that is exactly why I asked her to speak to you today, because she has so many wonderful qualifications. She's going to tell us today about equine metabolic syndrome. What is ID and how do we care and manage for it? So thank you. Dr. Adams, take it away.
Great. Thank you again, Carrie, for the invitation to join this great webinar series. It's good evening, everybody. I'm joining you from Kentucky. Hello, Karen. Hey, Ken, if you're on there. But as Carrie said, I'm going to focus in on talking about equine metabolic syndrome, but I really want to home in on insulin dysregulation, or what we refer to as ID. So what is insulin dysregulation? Why do we care about it? And really, how do we manage it when we're dealing with the equine metabolic syndrome horse? And so not only as the study of equine metabolic syndrome and insulin dysregulation a part of my research program at the Gluck Equine Research Center and a big part of my career, but it's also part of my personal story. As you can see pictured here is my own EMS case, little Taz, who he would like to not admit that he is, but this is just a picture that I thought I would share. But not only, again, am I passionate about this study from a career standpoint, but also from a personal standpoint. And so, you know, my goal with my program is, again, as Carrie mentioned, I maintain a herd of equine metabolic syndrome horses, as well as PPID horses at the University of Kentucky. And so the goal is, you know, how do we learn more about these equine endocrine disorders? How can we manage these horses better? And how can we learn more, again, about both PPID and EMS? And while I have PPID up here, I am going to pull in a little bit of talk about PPID, because PPID horses can also suffer from insulin dysregulation. And while EMS and PPID are very two distinct equine endocrine disorders, again, the central tie in this talk is the fact that both EMS horses and PPID horses suffer from insulin dysregulation. So I'm going to talk about, you know, I'm going to bring us back to equine metabolic syndrome, but I'm really going to home in on what insulin dysregulation is, how do we diagnose it, and then how to manage. And so why do we care about both of these equine endocrine disorders of EMS and PPID? Well, as our horses are living longer, the aging horse population is growing. And so you can see the numbers here up on the screen. You know, a significant percentage of the aged horse population in the UK, Australia, you know, the United States statistics are a bit lagging, but, you know, there's a significant number of these aging horses, and we really consider the aged horse over 15 years of age to be significant across the world. And in fact, from one of the recent USDA surveys, it showed that almost 50% of equine operations in the United States has at least one senior horse on the property. So we care about the aging horse population, and with the aging horse population, we see more numbers of horses affected by equine endocrine disorders of EMS, ID, and PPID. So the numbers are out there. And if we take it back to looking at specific statistics of numbers of horses affected by equine metabolic syndrome, we don't necessarily have those numbers. We know that, you know, obesity is a problem for the horse population. 20 to 51% of horses and ponies are considered to be overweight, obese, but we don't really specifically have the numbers of horses that are affected per se with equine metabolic syndrome or insulin dysregulation. And in terms of PPID, or pituitary pars intermedia dysfunction, which we used to refer to as equine Cushing's disease, prevalence, we have a bit more data on prevalence for PPID, showing that about 20% of the aged horse population is affected with PPID. And we think about 30% of PPID horses are probably affected with insulin dysregulation. So this is really why we care about equine endocrine disorders of EMS, PPID, and ID, because they are prevalent and they are part of our aging horse population. We also care about equine endocrine disorders, and probably the number one concern for all of us, I know myself, is the fact that these horses are susceptible and predisposed to developing laminitis. In fact, one fairly recent study has shown that laminitis cases present in a clinical study in which they studied the cause of these laminitis cases showed that about 90% of these cases had underlying endocrine diseases. And so we all know that laminitis is devastating. You know, it's basically, we're not going to get into the real specifics of what laminitis is, but it's basically damage to the laminar tissue that holds the hoof wall to the coffin bone that becomes inflamed, damaged, etc. And so it often leads to, unfortunately, euthanasia of the animal. And so that's really the big concern with, you know, having a horse that has equine endocrine disease, in particular the equine metabolic syndrome, in those horses that have insulin dysregulation. And I'm going to try to tie all of that together and why these horses are predisposed to developing laminitis. And I will say again that yes, there are a number of causes of laminitis, from sepsis-associated laminitis to support limb laminitis to trauma to, in fact, glucocorticoid injections. But endocrinopathic laminitis, which is laminitis associated with endocrine disease, is really the number one cause of laminitis based on some research that's been done in prevalence. And so let us I'm going to bring us back to equine metabolic syndrome. In fact, these are a few pictures of some of our equine metabolic syndrome horses at the University of Kentucky. I first want to talk about what is EMS. And so equine metabolic syndrome was first introduced to the industry by Dr. Johnson at the University of Missouri. He was the first person that really coined the term equine metabolic syndrome. In fact, this was almost 20 years ago. It's hard to believe. And since his sort of bringing this EMS to the industry, we've learned quite a bit about equine metabolic syndrome. In fact, a panel of experts in this field came up with a consensus statement in 2010, really defining what equine metabolic syndrome is. And so they came up with the definition of EMS as an obese, insulin-resistant horse or pony that has increased susceptibility to laminitis with really three main characteristics. They have increased adiposity, either regional adiposity or general obesity. They're insulin resistant and have predispositions towards laminitis. And so it was then this definition of equine metabolic syndrome has kind of changed with the more that we learn about these horses over time. And so then another group of experts, the equine endocrinology group, has now since changed the definition again, and they continue to update this definition of what equine metabolic syndrome is, the more we learn about equine metabolic syndrome. And so they define equine metabolic syndrome as a collection of risk factors that is associated with increased risk of hyperinsulinemia-associated laminitis, or HOW, which is essentially endocrinopathic laminitis. And insulin dysregulation is a consistent feature of equine metabolic syndrome, as well as increased adiposity. And while there are other factors that are present in equine metabolic syndrome horses, these are really the main driving factors of equine metabolic syndrome. That is insulin dysregulation and increased adiposity. And so you can kind of put that together in a picture schematic here. So equine metabolic syndrome, we now defined as horses with increased adiposity or general obesity, as well as being insulin dysregulated and predisposed to laminitis. So what is insulin dysregulation? Because you're all used to hearing the term of insulin resistance, right? So insulin dysregulation is any combination of a horse having hyperinsulinemia, postprandial hyperinsulinemia, or insulin resistance, okay? So when we talk about insulin resistance, we're talking about tissue insulin, reduced insulin sensitivity in the animal. And when we're talking about insulin dysregulation, we're talking about how that animal responds metabolically to consuming a meal or a high sugar challenge. And so a bit different in terms of being tissue insulin resistant versus having hyperinsulinemia. And so insulin dysregulation, again, is the key or central component or characteristic or problem that occurs in these equine metabolic syndrome horses. So again, insulin dysregulation is typically associated with the typical, as I've shown you pictures here, the obese equine metabolic syndrome horse. But I also want to make the point that insulin dysregulation can also be detected in lean or non-obese EMS horses. Insulin dysregulation can also be in the absence of equine metabolic syndrome and associated with PPID or equine Cushing's disease. So insulin dysregulation is not just per se an obese horse problem. And so when we talk about equine metabolic syndrome, we're talking about those horses that are, yes, typically obese and have certain genetic predisposition of being at higher risk for equine metabolic syndrome. So we know certain breeds are more predisposed to being EMS. But we know now that horses can also be lean and also be equine metabolic syndrome. And so, as well as being EMS with PPID. So I think what we're learning is that it's not just a problem for the obese horse. And so this is another diagram kind of showing you and really homing in on insulin dysregulation, that again, insulin dysregulation can occur in the obese EMS horse, but also the PPID horse and the PPID horse that's also EMS. So I think that's where, I think we need to kind of get, I guess, better at educating folks that insulin dysregulation is not just a problem for equine metabolic syndrome horses. And so here's a few examples of on your left is a typical equine metabolic syndrome horse that you would think of, with the crusty neck, the regional general obesity, who's also clearly insulin dysregulated. And then in the middle, you can see more of a lean type EMS horse that is also insulin dysregulated. And then on the right, you can see a PPID horse who you probably wouldn't guess to be insulin dysregulated, but is. So these are really the three categories in which are affected by insulin dysregulation. And that's really what we are concerned about when we talk about these endocrine disorders, because it's that insulin dysregulation that contributes to and leads to, and what we think is really the driving force to these horses to develop laminitis or endocrinopathic laminitis. And so just a quick side note, since I am bringing in PPID for those that don't know what PPID is, but PPID again is a problem of the aged horse. It's an age-related dopaminergic neurodegenerative disease that really affects the pituitary pars intermedia gland of the pituitary, and essentially that gland is not producing hormones correctly, which then disrupts an entire hormone signaling activation pathway in the body, which then leads to clinical signs of PPID. As many of you probably know, one of the number one clinical signs of a severe horse, or a horse that is affected with PPID showing severe clinical signs, is hypertrichosis, or a thicker abnormal hair coat that has slow to shed out, etc. But also, as you see in the red here, that these horses can be insulin dysregulated as well. So something, again, not to ignore in the PPID horse. These horses also experience muscle loss, weight loss, etc. And so, but I want to bring us back to insulin dysregulation. Why is insulin dysregulation bad? So insulin dysregulation is bad because fairly recent studies have shown that this prolonged hyperinsulinemia, or this increase in insulin in these animals, is what really is the driving force to set off laminitis to occur. And so while we don't know the exact mechanisms that are occurring in the hoof or the laminar tissue in the hoof, we do believe that it's insulin that is triggering this cascade of events to occur for the laminar tissue to undergo abnormal proliferation, which leads to elongation and weakening of the laminar tissues. And then you get basically the laminar tissues not doing the job and holding the hoof capsule to the coffin bone in the hoof. And so it's really the insulin, because studies again have shown that basically infusing healthy animals with insulin above normal levels of insulin, and basically making the animal hyperinsulinemic, those animals then go on to develop laminitis. And so we've learned a lot in this area, and which is why we are trying to really push the word that it's this hyperinsulinemia as part of this insulin dysregulation in the equine metabolic syndrome horse or the PPID horse that may have insulin dysregulation, and this is why it's bad. And so when we talk about laminitis associated with equine metabolic syndrome or PPID, again, we categorize it as, most of us say laminitis, but to be more accurate and correct, we categorize it as being endocrinopathic laminitis, or now we have tried to use the more appropriate term as hyperinsulinemia-associated laminitis, or what we call now as HAL. And so it's this high blood insulin concentration that damages the laminar tissue in the hoof. And so again, it's this insulin dysregulation that drives the development of laminitis in these animals. And so with that being said, prevention is key, right? So diagnosing insulin dysregulation appropriately and then managing it appropriately is really key in helping these animals trying to prevent laminitic events from occurring in these endocrine disordered animals of equine metabolic syndrome or PPID. And so the equine endocrinology group, if you just Google equine endocrinology group, and that's coming out of Tufts and really is organized by Dr. Nick Frank, but really a panel of experts that have come together and try to keep basically updating everything that we learn through research about equine metabolic syndrome and PPID, and how do we appropriately diagnose both conditions, and then algorithms for how do we manage them. So they're really, really great resources if you want to go ahead and have a look at those. But basically, at what point do you decide, when do I test for insulin dysregulation? Well, you're going to test or have your veterinarian test for insulin dysregulation when you have a suspect case, when you have an animal, horse, pony, donkey, that has presented before with laminitis issues, that has regional adiposity, that has general obesity, that's having the signs of being equine metabolic syndrome, or potentially even PPID. At that point, I highly suggest that you have your veterinarian work with your veterinarian to test for insulin dysregulation, as well as if you suspect, you also need to test for PPID. And so how we now have basically decided that testing for insulin dysregulation is best if you look at using dynamic tests in the animal versus just looking at rest or basal insulin concentrations in the blood. So two ways you can do this. So you can have your veterinarian pull just a blood sample and then look at the basal insulin concentrations in that animal, or you can dynamic test that animal by essentially challenging that animal with an oral sugar test, or an OST is what we call that test. And then basically after the oral sugar challenge in that animal, you're looking at the insulin response after that oral sugar challenge. And so when you're looking at resting or basal insulin concentrations in an animal, you want to make sure that those animals have not been grained. You want to collect these samples during the morning hours. You can basically pull the animal right off of a semi-dry lot, pasture, hay, etc. In fact, we recommend now that you don't fast the animal when you're measuring or testing for basal insulin. But when you're performing the oral sugar test, we've always recommended that you withhold or actually fast the animal for 3 to 6 hours. Again, the way that we're basically the best way to diagnose for insulin dysregulation is using the oral sugar test, because insulin dysregulation is going beyond the animal being tissue insulin resistant, and it's really a part of this equine interoinsular axis that plays a role in this hyperinsulinemia process that's happening in these insulin dysregulated animals. So you can look at resting or measure resting insulin concentrations, or look at insulin responses to the oral sugar test. Both are great ways to diagnose an animal for being insulin dysregulated. But again, a few points when you're looking at the resting basal insulin concentrations. Again, no fasting is needed. Hold grain if the animal is receiving grain. And then it's really important to have your veterinarian, send these samples off to appropriate labs. And one of the gold standard labs that we always recommend veterinarians use is Cornell University or Michigan State University, or some of the commercial labs like Antech are also great. They have validated assays. They have established appropriate reference ranges for what is basically not insulin dysregulated, what is an insulin dysregulated suspect case, and what is insulin dysregulation. So very, very important, and important that the samples are handled appropriately as well. So again, the EEG has put out the recommended protocol for the OST. And basically you're administering you could work with your veterinarian, administer the K-Row. Basically you're administering a bolus of light K-Row syrup to your animal and collecting blood about an hour, right at an hour after administering that light K-Row syrup. So again, shipping those samples off to a reputable lab that knows the reference ranges and the cutoff values for what an animal, for being a insulin dysregulated case or not is very, very important. But we had a couple of questions of our own just from having the herd of horses that we do, the Equine Metabolic Syndrome horses and the PPID horses, that, should we be using different doses of light K-Row syrup versus low dose versus the high dose, because that was still questionable, and what dose should we be using with this oral sugar test? And then really, should we be fasting or not fasting the animal when we use the oral sugar test? And then what's the effect of season on these insulin responses in these animals? And so I know we're quite a bit behind schedule, so I'm just going to quickly go through some of these little studies that we have done recently in my lab. So basically looking at, these are previous studies that have been done in metabolically normal animals, comparing whether you fast or not fast, and you're looking at insulin versus ponies, whether you fast or not fast. But nobody had really looked at this with the insulin dysregulated horses until recently. We just recently published this work where we wanted to look at the effective dose and fasting on the OST. So what we did is we did a couple of crossover studies where we gave high versus low doses of OSTs, and we had a fed versus fasted group. So this is just some insulin data. On the left are your non-insulin dysregulated animals, or the animals that are normal, metabolically healthy animals. They're not EMS, they're not PPID. So you can see that their insulin responses are low, which is normal, and that's what we want, right? And those horses are definitely not predisposed to developing laminitis, at least not yet. And then on the right, you can see the insulin dysregulated horse responses in terms of their insulin values are significantly higher across the board than the non-insulin dysregulated animals. So that's a nice visual for you. That is, those are clearly insulin dysregulated animals. But then if you look at the responses, and when we fed, basically pulled them off of pasture or forage, whether they were on a semi-dry lot or receiving forage, that would be on the left, or the FE responses. And the fasted, per se, we did a short fast of 3 hours where they received nothing on the right, or the FA responses. And what you can see if you look at the basal insulin, or the T0 response, so there was a difference. And if you basically pulled the animal out of their current housing conditions and just sampled the blood versus doing that fast. So fasting actually reduced the insulin in those animals. But if you looked at the T60 response in the black bars and how they responded after that oral sugar test, 60 minutes after that oral sugar test, really no difference. And then if we look at the low dose K-Row syrup versus the high dose K-Row syrup, in these insulin dysregulated animals, it did not matter whether you used high or low dose K-Row syrup. So again, basal insulins, be very careful if you're interpreting this data using a fasted horse, but the OST seems to not be affected whether the animal is fasted or not. And then no apparent really advantage of using that high dose of K-Row syrup. If you are going to use the high dose of K-Row syrup, it's very sticky. So then we were interested in looking at the effects of season on these OST responses and just insulin responses over time in these insulin dysregulated metabolic syndrome horses. So we followed these horses. We followed insulin dysregulated horses and non-insulin dysregulated horses over time, and we looked at their response to the OST, as well as body weight, body condition, cresty neck scores. And so we collected samples for 2 years during winter, spring, summer, and fall, collected all these measurements. And we also collected forage samples. And so what you can see in the ID horses, when we look at body weight, body condition, cresty neck scores across season, we didn't see any changes in these animals, which was interesting. We did see some changes in the NID animals. And we look at the nutrients from forage, we did see some changes in the pasture samples as expected, in crude protein and water-soluble carbs going up in spring, but in the forage and the hay samples, we did not see changes across time. But we did see changes in basal insulin, not in the NID animals or the metabolically healthy animals, but in the ID animals, we did see some changes in insulin, basal insulin, with an increase in spring compared to fall. And if you look at the OST insulin responses, again, NID animals, not any real big changes over time per se, but if you look at those insulin dysregulated animals, you see some significant changes with winter and spring insulin responses being higher than summer and fall. So season clearly has an impact on insulin responses in these animals. What's driving that? You know, there could be multiple factors involved, from the nutrients in the forage, pastures didn't quite make sense and match up with the insulin responses in this study, keeping in mind that all of these horses were housed on the same farm, under the same conditions, under the same management practices. So it was a very controlled study, but there could just be some metabolic shifts in these animals as we undergo different changes in seasons. So I think it's a caution to veterinarians to keep that in mind when they're performing these diagnostics and looking at insulin responses to, especially the OST, when diagnosing insulin dysregulation. So kind of in a summary with some of the insulin dysregulation work that we've done, fasting again does impact basal insulin, but no real difference in how fed versus fasted affects OST responses, nor is there a difference in high dose versus low dose in the oral sugar test, but season does impact these insulin responses. So we've published all of that work, happy to share it. One thing I wanted to bring up, not only is blood work, really the gold standard in diagnosing insulin dysregulation, but cresty neck score is really a good predictor, and research has shown that, of insulin dysregulation. And so there's a cresty neck scoring system that was developed in 2009. There's also a neck circumference measurement that one could use. But basically, this cresty neck score is a good indication of an animal being EMS, insulin dysregulated, if they have a cresty neck score above 3. So coming to management, because I know we want to leave time for questions. Basically, if you have an insulin dysregulated animal that's obese, you're wanting to reduce weight, so limit the caloric intake in those animals, reduce NSC in these animals. So all management practices, whether that's through dietary or through exercise, if the animal is an obese ID animal, if they remain obese and are basically resistant to weight loss, then you want to consider medical management of those animals. Now, if you're talking about a non-obese insulin dysregulated animals, they are very difficult to manage. But you still want to be able to manage them on a low NSC diet. But yes, because they're not obese, they may need a little bit more calories. So you might want to consider having increased calories by increasing the fat intake in these animals. So management definitely start with diet. And diet is very, very important. So you want to reduce body weight, body condition by basically lowering the percent of body weight that you're feeding to this animal. In terms of forage, they should be on an all forage diet, consuming low NSC forage, limit the pasture, if not completely eliminate the pasture. Multiple ways. Again, this is a nice nice, the EEG, Equine Endocrinology Group, has a few nice guidelines if you want to check those out, because I am running out of time. Also want to consider muzzling your animal, but don't muzzle for more than 10 hours at a time. Strip grazing, turn out during safe hours, overnight, early mornings, where the NSC levels are, we think, lowest, and test the hay that you're feeding these animals, test the pasture, because we want to definitely lower that NSC. Again, when you're talking about a lean equine metabolic syndrome animal, very difficult animal to manage, but again, manage similarly to the obese EMS animal, but obviously you're not wanting that animal that's lean EMS to lose much weight, but you want to put them on a low NSC diet. And also the EMS horse with PPID, depending upon body condition, is how you want to manage that animal. Exercise, so diet first, but bring along exercise, really important in helping manage these animals, and improving the insulin dysregulation in these animals through exercise. Again, check out the EEG. But when it comes to diet, we all say feed low at NSC to these animals, but we really don't have a handle with what that magic percentage of NSC that we should be feeding to these animals. And so that was some recent work that we have, we've recently published out of my lab as part of Eric and Megan's PhD program. We really wanted to understand how these insulin dysregulated metabolic syndrome horses are responding metabolically to different feedstuffs. So we designed a couple of studies and compared essentially how EMS insulin dysregulated animals respond after consuming various meals to non-insulin dysregulated animals. So we had 4 different diets, and we collected blood samples pre and post giving these diets. And so the diets were basically ration balancers, because that's how we want to maintain these animals on an all forage diet, and bringing along a ration balancer if the forage is not covering all the nutritional needs of that animal, right? So we gave ration balancer that had high protein, low protein, and then a mixed, as well as cracked corn as a nice positive control, keeping in mind we're only offering or feeding these animals one time with these different diets. So you can see the NSC is actually similar with the ration balancers, which was nice, but then we had differences in crude protein, but obviously the NSC for the cracked corn was the highest. So you can see in the left here is the non-insulin dysregulated horses and how they responded over time to the various diets. They didn't really respond much, which is normal, which is what we would like to see. But you can see on the right how the insulin dysregulated animals responded to the various diets over time, with a significant increase in their insulin post-prandial quite immediately after consuming these very small meals of ration balancer with high protein, cracked corn, ration balancer, low protein in the mix. And so we did some statistics to look at area under the curve. And so you can see that the cracked corn responses were the highest, but we're really, when we looked at the statistics, it was really just the cracked corn that was significantly different from the ration balancer with the low protein. So goes to show you that, protein is also kind of a driver in this whole response, as well as NSC. So we wanted to know, okay, given these ID horse responses, gosh, do they just respond to anything that you feed them, whether it's high NSC, low NSC, high protein, low protein? So we designed another study where we integrated and we came up with a low, very, very low NSC, of NSC around 6%. And we wanted to know, okay, would these horses respond to an NSC that's even that low? And so what we found, this is looking at the delta responses of the NID horses in the black to the various diets, and the ID horses in the gray to the various diets. And what you can see is in the middle that the ID horses had a very low, almost normal looking response to the various diets. So that kind of made us think that, gosh, it's just not a matter of feeding these animals that elicits this postprandial response, that it's actually the different components of the NSC being really the main driver in these responses. So we definitely can't rely on nutrition based on normal, metabolically healthy animals. And we do see that NSC really is the main driver in these postprandial insulin responses. But we really need to further validate where is the threshold for NSC, what is a safe level of NSC to feed to these horses so you don't get an exaggerated postprandial response in these horses. We are currently working on that. We just submitted a publication with that work, so hopefully we'll be able to share that soon. And that just feeding a meal does not induce an insulin response in these animals. So apologies for running through this, but I know we were quite behind in the schedule. But take home on all of this, that insulin dysregulation is not only a problem for the obese equine metabolic syndrome horse, but also for the types that are lean EMS ID horses and PPID horses. Diagnostics are critical. Don't just guess. You definitely need to work with your veterinarian and properly diagnose for insulin dysregulation, as well as if you suspect your animal or horse to be PPID. And not all obese horses are insulin dysregulated. Preferred method to diagnose insulin dysregulation is using the oral sugar test. If you use just basal insulin, interpret those results with caution. And I think continual monitoring of these animals with diagnostic testing is really critical, especially how we know that they change across seasons, which may make them more susceptible to developing laminitis across different seasons. And management is a team effort. So pull in your veterinarian, pull in your nutritionist, pull in your farrier, podiatrist. A couple of other considerations. If your animal is insulin dysregulated, I highly suggest to avoid corticosteroid injections. Be very careful if you do. And hoof care obviously is also very critical with these horses. So it takes an army to definitely do the research that we do. Thanks to my entire team. Takes a lot of money to support our research program, that's for sure. So many thanks to the sponsors, and a special thanks to Mars, because they actually supported the work that I just showed you. So with that, I will stop, and hopefully there's a few minutes for a few questions.
Yeah, we definitely will make time for questions. There's been a lot coming in. I know Dr. Brooks has been busy answering some of hers. So I just want to say one thing, and then I will go ahead and throw out some of the questions. So if anyone does have to leave, because we will spend a little bit of time going over time, I did pop, and I'm going to put in the chat as well, a link to a survey or an evaluation for our speakers and for the talk tonight. Even if you filled it out last week, you can fill it out again for just this section of talks. So please take time and fill that out, because it really helps us gauge in what people thought, what they might want to do different next year, and what they might want to see. So I do have some questions kind of right along the lines of how you ended, Dr. Adams, with some recommendations on diets. And actually one question is about how to get the NSC down in the hay. What are some soaking techniques? And is there any information on excess iron in the diets of these horses? Can you give any feedback?
Yeah, so good question. So testing your hay is always recommended if you have the ability to, ship off your hay samples to have them analyzed. I think that's really important, especially if you have access to a similar batch of hay that you're going to be getting. But if you don't have access and don't want to mess with shipping off your samples to be tested for NSC, a way to lower NSC is, yes, to soak in cold water for at least 30 to 60 minutes, and make sure you dump off that excess water and then feed your hay. But I think really getting basically, even if you have to use a body weight tape, get a proper body weight on these animals and really weigh out your flakes of hay and how much you're feeding these animals. And, you know, if you're wanting to induce weight loss, you really need to be feeding 1.5% body weight, start there. And if they're not losing weight being fed at that rate, then you could slowly, carefully drop that.
Yeah, so what about these older horses? Do you recommend getting routine blood tests done on them,
Yes.
regardless of clinical signs?
Yes, so I just think it's a good idea for the older horse as a routine, you know, veterinary exam once per year, twice per year, even better, to do an endocrine panel on them, just to get a nice base, you know, follow these animals over time and follow their endocrine status over time. It's yes, I think it's a great tool to implement into your program for your older horse, for sure.
Yeah, a lot of questions. I'm actually starting to answer them as well, but since there's so many of them, I'll just ask you to do it live. About NSC. I've said, you know, non-structural carbohydrates include sugars and starches. But I know, you know, you read everything online and some say, just look at sugar, others say, just look at starch. Can you just clarify so everybody knows?
Well, the work that we just submitted for publication, we actually fed basically titering levels of just sugar and titration levels of just starch to better understand what is the driver in NSC that's causing these exaggerated insulin responses. Is it starch or is it sugar? And so with this recent work that apologies that I can't share that because it's actually just came back with revisions. Yay, it's been accepted, but not published yet. We will have those answers to tell you whether it's sugar or whether it's starch that's driving these insulin responses, but both do play a role.
Thank you. What about breed specificity? Is there a difference in breeds with basal levels of insulin?
So yeah, so you know, breeds play a role in predisposing some of these animals more so to being metabolic syndrome or insulin dysregulated. But to be honest, I've seen even thoroughbreds become insulin dysregulated. So, you know, within my herd, we are mixed breed. And interestingly enough, we do see pretty not real great variation across the horses of different breeds. Especially when they're quite severely affected by insulin dysregulation. So I don't just say, okay, well, your horse is not a thrifty breed, you shouldn't worry. I would not have that mindset because really insulin dysregulation can affect really any breed if environment and management is not appropriate.
And a couple questions along the testing lines, testing for, you know, ID or Cushing's. I know you mentioned 3 to 6 hours fasting, and I know a lot of people are saying, oh, they've been told 12, and what happens if they change now to 3 or to 6? And also with seasons, does it matter what part of the country you're in, whether you're in Canada versus Kentucky, et cetera?
Yeah, so in terms of the fasting, like the research I just showed you, we've found that fasting, you don't necessarily need to fast, but I do think it's important to be consistent with the method that you're using with your horse. So if you're wanting to follow that horse over time, I would pick one method and go with it, but I would highly recommend too what works best for your scenario. Some people may not be able to do a 12-hour fast, but really research has shown, and I've just shown you, that fasting is not necessary. A short fast is actually a little bit better because then people start to worry about, you know, a horse shouldn't really be fasted for 12 hours, not good on the GI health. So you might think about transitioning to a shorter fast and then be consistent with that method over time. So 3 to 6 hour fast, or based on our research, fasting really doesn't seem to make an impact on the OST responses. It did with the basal, but and then in terms of season and time zones, you know, I think I think that research needs to be done. You know, we obviously were just looking at temperate climate of Kentucky and how changes occur with season, because we don't really understand fully what's driving those seasonal changes. So what's happening in Kentucky seasonally may not be happening in other areas. So I wouldn't just can just guess that it's what we saw is what's happening in Canada. You know, it would be difficult to say.
Mm-hmm. So one more, and I'm trying to kind of combine themes of questions here to take less time, but let's try one more before I throw some at Dr. Brooks. But so you were mentioning about the fasting, and we know that that can affect stress levels as well. So I'm assuming just the stress alone and the fast can possibly affect the insulin as well, right? Yeah.
I mean, that's the idea of what's probably going. I mean, part of what is going on when these animals undergo a fast, at least for a long period of time. Yeah.
And what about using these oral sugar tests for horses that are, you know, possibly laminitic or have laminitic?
So if they're actively laminitic or in a current laminitic episode, absolutely no, do not use the oral sugar tests. But we have had no issues, and we have performed, I don't know how many hundreds of oral sugar tests in these equine metabolic syndrome, insulin dysregulated animals, and we've had no issue with those horses developing laminitis because it's a bolus. It's essentially, the oral sugar test is essentially mimicking a meal or a horse consuming even, you know, not even just a meal, but what's mimicking in their, you know, what's happening when they're out grazing on forage or pasture, but just a little bit more sugar in that. But it's a short bolus where you're not giving it over time. And that's really what we think, you know, is setting those animals up for developing laminitis, is it's that prolonged hyperinsulinemia response over time that triggers that laminitic episode. So yeah, I would not worry about that.
All right, I'm going to bring in Dr. Brooks as well, because we do have a couple. There was a lot of Arabian questions, so thank you for answering some of those in the chat. But I think there was a few that didn't get answered, and one of them actually deals with, and I've not actually heard of this, so I really want to know the tarpan, TARPAN, and the influence of the tarpan in the Arabian.
Right. So the tarpan is a primitive breed that was attributed to be the European wild horse, and is essentially extinct to date, although there are some groups that are trying to recreate it from some populations that may still carry some of that ancestry. That recent project that looked at the two regions of the genome that changed so drastically 3,000 years ago did examine some tarpan-like ancestries compared to some modern groups and some museum specimens of likely tarpan origin. And those animals, very interestingly, had a mix of that old domestication ancient lineage and the new modern horse lineage. So they seem to have sort of spanned that changeover into the new modern type of horse. We don't see evidence for that old tarpan lineage in the modern Arabian horse. So if they contributed, they probably didn't carry traits that would have perpetuated their genome in big percentages in the modern Arabian. Now, that being said, we do have some very interesting research underway looking specifically at some of our Arabian genome sequences compared to some of those ancient genome sequences. So I hope to be able to tell you more as some of that research begins to get us new results.
I guess kind of along the same lines, could the relationship between the thoroughbred and the racing Arabians be due to the contribution of the Godolphin Arabian?
Yeah, we get that question a lot because, you know, whenever we talk about this, people go, oh yeah, of course, because the thoroughbred is actually from pre-Arabian stallions. And I wish we had evidence that that was in fact the case. But there are two key pieces of evidence that we have so far that point to a modern origin of much of that Arabian genome, a thoroughbred genome in the Arabian, rather than an ancient one. One piece of evidence comes from that Y chromosome molecular clock, in that we know that the Y chromosomes that entered the Arabian populations came from animals that lived after 1800. And that is long after the Godolphin and Darley sequences. Like, we can see changes that happened over time. The other thing is that, when we look at the size of the individual pieces of the genome, the length of those pieces tell us how long it's been since that admixture event occurred. And those lengths tend to tell us that many of these admixture events were, of course, recent. Now, the size of the pieces for those Turkish and the sort of the chocolate brown ancestry that you can see mixing into the thoroughbred, those probably are ancient. And it looks like the data that we have support that in that case, we likely have signatures of those animals, the Byerly Turk and the Godolphin and Darley Arabian. What's interesting, though, is that when we think about the romanticism of history, we probably over attribute the total contribution of those historic stallions to the modern thoroughbred than the proportion of their genome that actually still persists there.
Good stuff. And I guess one last question for you, and then I'll ask maybe one or two more for Dr. Adams before we cut it off. But do you recommend a particular service provider for DNA or genomic testing for either breed or health conditions?
Yeah, and I see some questions that have come in about using some different services for that. Now, first of all, I have to say that as a state employee for Florida, I am not allowed to specifically recommend any one testing service. So I'm actually pretty impressed that Dr. Adams got to call out on the excellent services at Cornell. I'm not allowed to do that. What I can tell you are some guidelines. And when you're talking about ancestry, since I see a question specifically addressing ancestry testing, make sure that the service you use utilizes thousands of markers called SNPs and not dozens of markers called microsatellites or classic parentage testing. The numbers on the microsatellite test are not large enough to get accurate results, even though those are marketed as such. You need thousands of SNPs to do this right. And I won't call out any one service provider. I'll just tell you that's what to look for. You also want to look for laboratories that participate in the International Society of Animal Genetics comparison test. So that's a volunteer process that's run every other year, but it does say that that laboratory is an important part of the international network of testing labs. Doesn't, again, guarantee results or give you any indication of quality. You also should look for providers that will reference the published research, because there are a number of providers, especially in the sequencing area of techniques, that are starting to offer tests that are not published, that are completely unvalidated, and some of those are total garbage and are the bane of my existence, because people say, oh, I sent it there and it didn't work, therefore genetics is all garbage. And I'm like, no. There's just a few groups that are overstating their findings, I will say. So make sure they're using peer-reviewed published literature to back up their tests. Finally, look for good value. It is technically feasible to do thousands of SNPs very cheaply. They don't need to charge you $25 per marker. And look for good quality customer service, because the outfits that have the time and knowledge to talk to you are the ones that also put a lot of work into their R&D and into providing a good quality assay. That was a lot. That was like 5 separate tips, but that's the best I can do.
I understand. I do the same thing with nutrition, is can't do companies, but can give you a lot of other recommendations. So, which, here we go. My next question is going to be all on companies, and it's thrown to you, Dr. Adams, about, okay, we know Presend is the one pill that we use for Cushing's. Is there anything else on the way? And then someone also asked about levothyroxine for ID in horses. Can you comment on any of those?
Yeah, so no, pergolide or Presend is, yes, the gold standard treatment for PPID and the only treatment that is regulated on the market for PPID that I'm aware of. There's nothing, there are a few things in the pipeline, but it'll be quite a while before, sufficient research is conducted on those products. But Presend is the gold standard treatment for PPID at this time. Levothyroxine or Thyroid L is a medication that is used to try and help induce weight loss in those animals that are weight loss resistant with diet and exercise. So if you've tried diet restriction and exercise in your animal and you're still having obesity problems, you will work with your veterinarian and consider using Thyroid L, but only use it for a short period of time. Thyroid L is not meant to be used for a long period of time to treat these animals. It's merely to try and help them lose weight, thereby hoping to improve insulin dysregulation in these animals for a short period of time. Then you can slowly taper them off of Thyroid L and then manage them at that proper body condition with diet and exercise. I'm not sure what the question was around Thyroid L, but yes, that is an option.
Yeah, if you recommended it.
That's right. I'm not recommending it. I'm recommending I start with diet, dietary management and changes, as well as exercise. And again, if that doesn't work, then consider alternatives such as Thyroid L, but definitely work with your veterinarian on all of that.
Yeah, you just mentioned short term, and I know someone said, what do you mean by short term for Thyroid L? Week?
3 to 6 months. Yeah.
Thank you. Well, I know there's questions we haven't gotten to yet. I'm very sorry about that. But just to allow our speakers at least a little bit of time to themselves tonight, I think we're going to cut it off. I will say you'll get an email tomorrow thanking everybody, also providing you my email. If you do have a specific question for the speakers, please go ahead and send me the email and I can forward it to the speakers. And I'm sure I will ask them now if you would be willing to answer some of the questions that we might not have gotten to. But I really want to give you all a huge round of applause. I learned so much today. The talks were great. Thank you. And thank you all attendees very much for joining us. I think it was a great night. We've got one more next Tuesday. Join us for more talk on nutrition. It seems like nutrition was extremely popular in the survey responses and should be a very popular night as well. So with that being said, please take time. I'll pop in the survey one more time for everybody to please take it. We really appreciate any feedback. But thank you again, everybody, and thank you, speakers.